you. So this will be a little bit different talk. So I'm going to be talking about a type of gene-based vaccine. So you heard a little bit about using things like plasmid vaccines, and you guys are probably familiar with, the concept of using a gene as a vaccine rather than the protein itself. So what I'm going to talk about today are a set of viral vectors. So I did a postdoc with Stephen Johnston on using things like gene guns for vaccination, and at a certain point, as we started working on infectious diseases, the fact that most of these things come in at mucosal surfaces led us to try to find good ways to actually educate the mucosal immune system to try to set up a mucosal barrier for infection for these infectious diseases. And so, a lot of different systems could be used, but, if you use a virus to deliver genes, you're sort of letting evolution do the heavy lifting for you. These guys have already evolved to put genes into cells and express them. So many people are using these to deliver antigen and vaccine genes. So what I'm going to talk about today is using adenoviruses as vaccine vectors, and so this is probably familiar to a lot of you. Basically, if you play with a lot of different viruses, put them in vivo, adenoviruses are arguably one of the most potent ones at delivering genes in a living animal, and so they can be quite potent for a variety of applications. So these are viruses that could be amenable to translation as human vaccines. In fact, one of the two Ebola vaccines that is now currently in testing is an adenovirus vaccine. So these are icosahedral non-enveloped viruses, so they're very stable. You can lyophilize them, you can freeze dry them, and ship them with minimal cold chain in some cases. They have a very large DNA genome, which means you can put a lot of genes into them. And unlike certain viral vaccines, there are actually at least 60 serotypes, different immunologically variant versions of these viruses in humans, and there's even more in other species. So that means that they're not necessarily going to be a one-off vaccine. If you use it once, you could use a different serotype and evade immunity to the first one you used. And then, as I already said, they're pretty potent in vivo for gene delivery. So, if we looked at a wild adenovirus as a gene delivery vector, this just shows sort of the genes that these adenoviruses have, a replication-competent adenovirus. So you might put your favorite gene into this to use as a vaccine. And if you're using this, and actually some of the vaccines that humans have gotten are live adenoviruses given by pill. So the way these work is they would go in, they'd infect cells in the body, and then in this case, if one virus got into the cell, it would deliver one copy of its own genome along with the vaccine gene that you wanted to carry. So once here, though, then they start to take off. They replicate their genome up to 100,000-fold in that cell, and so you get this advantage that that one gene you put in gets amplified thousands of times, so you can also amplify the amount of the antigen you're producing in the vaccine. The problem is that when these amplify their DNA, they also amplify themselves, and so you can create basically a wave of infectious viruses that can actually cause an adenovirus infection itself. And, so the question is, if you use this vaccine for whatever disease, will you may protect against one thing but cause an adenovirus infection in the person that got the vaccine, the nurse, or maybe the family members who took the kid home. So the vast majority of adenovirus vaccines that are out there are actually what are called replication defective adenoviruses. So these are deleted for the E1 gene, which is a pivotal regulator of the viral life cycle. And so this makes these vectors very safe. So they go in, they infect a cell the same way, but once they deliver their gene, they don't amplify it. And so this means they're very safe. You make no infectious adenoviruses, but they're also arguably fairly weak because you don't get the benefit of gene amplification. So one of my students worked in the lab to try to, get around this problem of these replication defective vectors can be pretty weak when we translate them. From back in the days of using plasmids and gene guns, we could protect the heck out of mice pretty well, but once these little things got translated into bigger and bigger animals and humans, they've become less and less effective. So this is another reason why we've gone towards trying to get a more robust platform to vaccinate with. So, looking at the adenoviruses, basically we were working on could we amplify the genes but also get rid of the danger of causing an infection. So we developed these things called single-cycle adenoviruses, and they're basically are viruses that will infect. They'll still amplify their DNA as well as the genes they're carrying, but they've been engineered so that they can't create any infectious adenovirus progeny. And so my student tested a number of different approaches to this, and we found one that seems to have a sweet spot for doing this. And so this is basically to knock out a gene in the virus that's involved in the life cycle after DNA replication occurs. And so that gene happened to be the best one so far has been this gene for a protein called 3A, which is a cement protein in the virus. And so to make a long story short, basically the student knocked out the gene for 3A in a low seroprevalence adenovirus called AD6. And then she deleted this and then made a complementing cell line that would provide that protein in trans so that you could rescue the virus as a vaccine. And so the initial test drive of this, what she did is she made a replication defective version, a replication competent version, and a single cycle version. All of these, in this case, expressing green fluorescent protein luciferase fusion protein, because this is it makes it easy to track things, but they can also serve as sort of a model antigen to look at antibody and T cell responses. And so if you take these three viruses expressing GFP-LUC and you put them on human cells in vitro, and then you expose them for 2 hours and 24 hours, and you quantitate the number of viral genomes in the cells with the replication defective, the single cycle, or the replication competent, basically if you give them 24 hours, the replication defective vector doesn't amplify the gene, but these other two do, and the single cycle vector replicates the DNA about 10,000-fold to the same degree as the whole replication competent virus. So, the problem comes if you have a vector like this is what animal model would you use, because you have to, your vaccine has certain biological requirements to go through its life cycle, and you also have pathogen needs for the animal model. So for us, we really only need an animal model to replicate the DNA to test drive the system. So, we could look at mice. Actually, Syrian hamsters are thought to be the kind of the ideal small animal model for adenoviruses. Ferrets are good models, and then, of course, rhesus macaques. So we've kind of played in these different models with different systems. So if we look at transgene amplification in cell lines in vitro with just comparing the replication defective and the single cycle vector, in this case we're just looking at transgene production, luciferase activity. If we look in human cells, you see about 100-fold amplification of protein expression by the single cycle. In macaques, you see similar amplification. In hamsters, you see amplification. Mice. And actually, in ferrets, it's actually only minor amplification, but there's still detectable change. So it seems like we could test these vectors in different systems. Now, that was a log scale. So if you look at this on a linear scale, you can maybe appreciate the sort of bang for the buck in terms of transgene expression by allowing the adenovirus to amplify the genes. And so this may be helpful in terms of translation. So if you take these vectors and you take advantage of the ability to image luciferase, if you take replication defective ad and inject it intravenously in mice, you can image some luciferase activity in the liver. But if you use the single cycle, you can see that it's amplified quite a bit. If you go intranasal, which is more what we're interested in terms of a mucosal vaccine, you can see a little bit of expression in the nares of the mice, but if we use a single cycle vector, you can see the nares are lit up, and also you see expression deep down in the lungs. And so if you go into hamsters and do something similar, it's a little harder to do luciferase imaging because these guys are big. These are about 125-gram animals. But you can see whether it's single cycle or replication competent that the amount of expression you get in this mucosal tissue is quite a bit higher. Now, when we then looked at immune responses against these reporter proteins, in this case GFP, so here we did single intranasal immunization in hamsters, and then we're looking at antibody responses in the serum and in vaginal washes over time. So this is one immunization. So if you look at PBS, you get background levels. If you do a replication defective ad, you get a decent antibody response to the reporter protein that is pretty stable. But when we use the single cycle, we see that the levels in the serum are markedly increased. Now, surprisingly, when we did the replication competent vector, it was actually no better than the replication defective vector. And this is in a model that supports adenovirus life cycle. Now, what was, for us, trying to make mucosal vaccines, it was more interesting that when we looked in vaginal washes after single immunization and looked weeks after single immunization, the replication defective gave a little bit of activity, but then the single cycle vector, one immunization, we saw the antibodies rise over about 12 weeks and stayed up for about a half a year. And the replication competent, again, didn't give much of a response. Now, we didn't expect this. We thought that at best the single cycle would be as good as the replication competent vector. And similar things have been seen sort of with things like other types of single cycle vectors for like hemorrhagic vaccines. But one possibility is that actually how these vectors interact with the immune system in vivo may be quite different than what we see in vitro. And one area is that as viruses come in, they can activate interference-stimulated genes. And so, and these have been shown to affect resistance to adenoviruses. And so just as a kind of a preliminary study, we compared these different vectors in interference-stimulated gene reporter cell lines, and basically we saw quite a bit of difference in stimulation of these ISGs by the replication competent vector than the single cycle. And this may have something to do with why we're seeing this be more potent. And so that we then went in single sublingual immunization in macaques with control and looked at antibodies in the serum. We saw not much going on here. The replication defective vector, single sublingual immunization, we saw a little bit of activity. But then the single cycle, we saw basically something similar. The antibodies went up over time until we stopped measuring them in the macaques. So we published this about a little over a year ago, so we've been plugging a bunch of pathogen genes into this now to see if it actually works. So, and basically focusing on just comparing the benchmark replication defective vaccines to these single cycle ones. So in one case, we put HIV clade C envelope and looked at different routes of immunization. So if we immunize by muscle or intranasal routes, these can be quite potent at driving antibody titers against HIV envelope by these routes. And when we've gone into hamsters with looking at anti-influenza antibodies, and again, this is single immunization, looking 6 weeks after one immunization, if you put different amounts of the vaccine intranasally, the replication defective vector generates good hemagglutination inhibition titers at different doses, but when we put the single cycle in, we get quite a bit more bang for the buck in terms of the level of functional antibodies against influenza virus. And we can, with a low dose, we can basically match the replication defective vector at all doses. And, you know, these are not great models for influenza. So we did do a pilot study in influenza and ferrets. So this is single immunization. And then if we now challenge these with influenza intranasally and then look at PBS or control single cycle vector and look at titers in the lungs days after immunization, days after challenge, that the single cycle vector is able to mediate pretty good protection after single immunization. So we think this is pretty promising, and so we're looking at a lot of different antigens now. And, you know, one kind of implication maybe is, you know, it's hard to extrapolate what will this do in human cells. Arguably, these human adenovirus vaccines are probably not working as well as they could because we're doing the tests in non-human models. A mouse or a hamster is still not as good as a human, probably, for hosting an adenovirus life cycle. So, you know, if you take primary human airway epithelial cells and you compare the vectors, so you put different amounts of replication defective ad on and different virus particles per cell. With the replication defective ad, as you go to less and less virus, with the GFP expressing vector, you see less and less GFP expression as we go out to this end. If you go in with a replication competent vector, you can basically transduce cells all the way out to 100 virus particles per cell. And if we look at the single cycle, you know, it's basically the same. So we're basically talking about a range of could these vectors actually, you know, confer the same sort of expression as the benchmark replication defective vectors using maybe 33 times less vector. You know, and if so, that may mean that one cell factory or one production of a single cycle might actually get you 30 or 100 times as many doses as compared to what's out there now. You know, so to summarize, these single cycle vectors seem to be pretty potent for expressing antigens. Testing them in animal models may kind of underestimate their utility, and we think they could be promising, particularly for mucosal single vaccination. And the person that did most of this work's Katie Crosby, and our collaborators are shown here. Thanks.